I really appreciate the opportunity to come here today and really speak to you as someone that's approaching this incredibly challenging and humbling disease from a perspective of virology and a background in antiviral drug discovery. And I'm just going to finish at the end in this talk focused on treatment of thinking about some of the future prospects, things that we might try to do to have an impact on this disease. So I just put these statements up in the beginning. These have been stated, but how one, in thinking strategically about developing a treatment, has to face this sort of conundrum between a rare disease and a common infection. And the numbers were pointed out, at least 50% or more of patients have been exposed to JCV, yet it lives in relative harmony in our bodies until the immune system gets tilted out of balance, either by immunosuppression or immunomodulatory drugs. And then we also know that the virus that's associated with the PML has undergone from rather profound genetic changes, in my view, very significant changes in the regulatory region of the virus, and also mutations in the coat protein that mediates its interactions with cells. And as stated, and I'll make the point again, and Dr. Major made the point this morning, we really don't understand very much about the steps that are involved in that transition from a benign, relatively benign virus to a disease-causing virus, and the pathogenesis of the disease. I think about JCV and PML research in three pillars. One just focused on the basic biology, a second one that's really focused on risk stratification and new tools to enable earlier diagnosis, and thirdly, approaches to therapy. And I'm a biologist, but I have to say that my bias is that we cannot underestimate the criticalness of the basic biology itself. It underlies everything else. And until we make inroads on that, it's going to be difficult or challenging to make further advances in the other two areas which we're engaged, which is risk stratification and therapy. At Biogen, it's a comprehensive research program integrating knowledge and resources, not only in internal programs, so there is a substantial effort there, but also in supporting academic investigators and participation in the consortium, which you'll hear a bit more about tomorrow. And then the goal of all of this is to provide physicians and patients with tools to better assess the risk, to diagnose as soon as we can, and should disease develop, to improve the outcome of that process. And it falls in a variety of areas that all require good understanding of the biology of the virus. Coming at this from an outsider, this is my own view on some of the big challenges in JCV research. One is, and some of these are points that Dr. Koralnik just made, it's just the difficulty of actually propagating the virus in the laboratory, having the most relevant cell systems. We don't really have oligodendrocytes, for example, that we can readily culture in the lab. It's very difficult to grow isolates from the brain, the pathogenic isolates. There's a laboratory strain that most people work with. The lack of an animal model. We don't really understand the reservoir of the virus in that period of time where we don't detect it and it's not active. We don't understand the circumstances that support transmission to the brain, and we don't know when and how these mutant viruses arise. So there are a lot of unknowns that make it difficult then to go in and be very strategic about thinking about therapy. And so I like to think of it as our toolbox it's not empty, but it's relatively light. And what we would like to have is substantially more fleshed out toolbox to work with. We've already heard about risk stratification. I won't repeat these 3 points of things that we know about risk, but what we would like to do is take it to the next level. And certainly this appears to be a multifactorial disease. We want to understand better about the host factors that are involved, the parameters of immune function, as Dr. Major talked about this morning. Also to analyze, is there any host genetic predisposition towards risk, viral factors that we've mentioned, and something that I'm personally intrigued with is any role of co-infection. What we do know about treatment is that for Tysabri-associated PML, the current standard of care is removal of drug. One can accelerate the removal of drug by plasma exchange or immunoabsorption. And this graph that I'm showing you shows the scientific representation of that. In the blue line, you can see that when after plasma exchange, the level of Tysabri drops. It drops quickly in a matter of days, and it stays down. But also, as we're going to hear about more in this session, there's the complication of iris. And the one thing from scientifically, what iris tells us is that the cellular immune response has been restored. It's come roaring back a little too strong, but it does say that the drug removal correlates with the return to the cellular immune response. What we've done also in this graph is look at anti-JCV antibody levels in the same period of time, and what you can see in the red line is why JCV antibody levels go down in short term with plasma exchange. Those levels return, indicating the humoral response can recover as well. And Dr. Kraunik went over some of this. I'm just looking at it in a slightly different way as some of the things that have been done. There's been efforts in immune modulators, such as an interferon, which is designed to program an antiviral response. That's an innate immune generator. And there was, I put inconclusive. I think Dr. Kraunik's conclusion was more accurately stated. There's also some notion about using certain cytokines that are immune boosters, either alone or in combination, but we don't have anything about that. This idea about finding a marketed drug that might work on JCV was a good idea. I call it the getting lucky approach. If you could find something that was already out there and had a safe profile, it could accelerate a development path. But this has proven to be hard in our own work at Biogen, where we screened over 3,500 marketed drugs, and this antiparasitic drug had the best in vitro profile. So we took it forward, but as you've heard, the results were not, we could not see any impact on the clinic. The notion of broad-spectrum antivirals, which is cidofovir, and it's the next generation of cidofovir, which is CMX. These are a class of molecules that have activity against nearly all DNA viruses, but we really just don't have any clinical data from them yet. And then where one would traditionally go in antivirals is to pick something that's really related to the JCV life cycle, and we don't really have efficacy on that either. Among the challenges is the preclinical package, which I've talked about, and especially the absence of an in vivo model. Typically, one would go forward with a preclinical package that had some assessment of pharmacokinetic, pharmacodynamic interactions. And without that bridge of an animal, you really cannot do that. You have to take the in vitro data and use your PK and assumptions about blood-brain barrier and model dosing to go in. That makes it difficult. The clinical scenario is very challenging. It is an acute, life-threatening infection. We don't know how big or small the window is with which we can intervene. I can tell you, coming from a background of influenza, for example, where one has 2 days after which you've lost your opportunity, it's not going to be that tight, but it may be months, it could be weeks, and we just really don't know this until we have some things at our disposal. The disease course is variable, and of course, as we heard, I think the number one challenge is the rarity of the disease. It makes it very difficult to plan trials. And this is just my encouragement to the regulatory authorities that we don't know really what is an established path for approval. There aren't really established endpoints or precedents for a registration trial. I just wanted to point out some basic virology here, and looking at the successes of drug discovery in other viruses, that there's a lot of potential points of intervention. Basically, any point in the life cycle of the virus is potentially a target, and there are a variety of successes out there from the various early steps of infection called entry. You can inhibit with target the host receptor, There are monoclonals now being used against viruses. Um, some viruses encode enzymes such as a protease that they need to process their own proteins. That's been a very good target in hepatitis C and in HIV. Where we have the most successes, though, are things that inhibit genome replication, so viral or cellular factors involved in replicating the genome. So we can take some lessons from this history of drug discovery and virology and see where we might make inroads in JCV. I won't spend too much time on this, but there are a variety of ways that one can approach drug discovery, including the more traditional approach of having a target-based, reconstituted enzyme protein in a tube, all the way up to blind infectivity screens that you could do in a whole infection type of approach. If you do the latter, then you have to take your chemical matter and backtrack and deconvolute what the target is. But all of these things are possible, and then we're in biologic era, and monoclonals is a very active area in infectious diseases using antibody-based approaches to treat viral diseases. I'm now going to tell you very briefly about two early programs, and we did want to come forward, even though these are early programs, and tell you about some of the things that we're doing at Biogen. The first one is about a program around neutralizing antibody as a potential therapeutic agent. We know that antibodies work by recognizing key determinants on the virus and prevent the interaction or the infection of cells. And the molecular hypothesis is that we can find a high affinity, potent anti-JCV monoclonal, neutralizing monoclonal, and we would have to deliver that at sufficient concentrations that it would reach the brain and slow spread and reduce the burden of disease and have an impact in the clinic and the prognosis. This is just some data from our program. We do have a collection of fully human neutralizing antibodies. They have excellent biochemical properties. The thing an antibody engineer would tell you that they look developable. We've learned, as has been seen in other viral systems, that affinity and potency don't track in a linear fashion, that there's often a threshold above which you don't see an impact on infection, but below which you will begin to see that, and we've seen that with our second set of engineered variants. And we know, and this is crucial, that we need to be able to neutralize a wide range of isolates, not just MAD1, but isolates that harbor these VP1 mutations. And that's going to be something that's very critical in advancing this. A second program I just want to mention to you briefly, T antigen is the only protein encoded by JCV that has an enzymatic activity. JC doesn't encode its own polymerase, it doesn't encode a protease. So the only protein that we can see that the virus encodes that has a specific activity is T antigen. It is involved in replication of the genome and therefore falls in this class of frequently successes. It has a helicase function and an ATPase function. And here the hypothesis is that if we inhibit the helicase or the ATPase function of T antigen, that this will block virus replication, and again, that this will be associated with reduced neurological damage and improvement in clinical outcome. We've done target validation on this by showing in these two graphs on the left side that if we mutate the helicase or the ATPase, in this case, binding site, then we make a dead T antigen. So we know the virus needs that ATPase activity for replication. We have completed a reasonably comprehensive high throughput screen on helicase activity, and this is just a little bit about our flow chart where we're looking at our structure-activity relationship to better advance our chemical leads. So to summarize, Biogen is committed to a very comprehensive collaborative program around basic biology, risk stratification, and treatment. That currently immune reconstitution, as we'll hear more about, is the standard approach in treating PML. There are some ongoing efforts in what I'm calling these broad spectrum antivirals, the cidofovir derivatives, and there may be others that we don't yet are aware of. But one we would like to see the field goal are very specific therapies that is, of course, safe and efficacious. We know there are challenges with regard to the clinical design and the logistics, And we very much feel that addressing these challenges is a global problem, and that's why we're all here. It requires a focused, concerted effort, a consortia of academia and industry, and certainly guidance from the regulatory authorities. So thank you.